Hello everyone, my name is Mackenzie McManus and I am looking forward to sharing with you the research I performed this past summer, where I was studying how the PI3 kinase AKT signaling pathway regulates IL-8 secretion from MUC2 mucin-producing colonic goblet cells. To begin with some background information, colonic goblet cells are specialized secretory epithelial cells present within the colon. These cells contribute to innate immunity through the release of mucins, which are building blocks for gut mucus, as well as through the release of cytokines and chemokines, which induce inflammation. As mentioned in the previous slide, goblet cells can induce the release of pro-inflammatory molecules. Such a response is regulated by signaling pathways. The pathways known to be involved include the IKB NF-κB pathway and the PI3 kinase AKT pathway. This research will focus specifically on the PI3 kinase AKT pathway. Keeping in mind these characteristics of goblet cells, what currently remains unknown is if mucin production in goblet cells impacts the release of pro-inflammatory molecules through the intracellular pathways, or essentially, does the presence or absence of mucins change the pro-inflammatory response? This research strives to address this gap. First, we hypothesize that high levels of mucin production by goblet cells cause activation of signaling pathways that then regulate the release of pro-inflammatory molecules. As such, the aims of this research were to determine pathway activation and to determine the signaling pathways that are specifically involved in pro-inflammatory regulation in cells with and without mucus. For the first aim, wild-type cells with high mucin secretion and MUC2 knockout cells lacking mucin secretion were interrogated with soluble amoebic proteins that were obtained from the parasite Entamoeba histolytica. Western blots were then performed to identify the activation of the PI3 kinase AKT pathway as a result of the interrogation. So when we look at pathway activation, we are looking to see if there is phosphorylation in the pathway. Based on the Western blots indicated in this slide, we can see temporal activation between 2 minutes and 1 hour for both wild-type and MUC2 knockout cells, and this is indicated by the increasing band strength seen here. As a result, we can state that the PI3 kinase AKT pathway was activated in both wild-type and MUC2 knockout cells. For the second aim of this study, both wild-type and MUC2 knockout cells were once again interrogated with soluble amoebic proteins from Entamoeba histolytica, but they were also interrogated with a PI3 kinase pathway inhibitor. The theory behind this is if this pathway is inhibited and it results in a reduced pro-inflammatory response, we can therefore understand that this pathway is involved in regulating the pro-inflammatory response. And so we used qPCR and ELISAs to quantify the amount of pro-inflammatory molecules that were produced on a transcriptional level and on a protein level. We used interleukin-8 or IL-8 as a measurement for the pro-inflammatory response. So based on the results indicated here, we can identify that when an inhibitor is present in wild-type cells, it results in a decrease in pro-inflammatory molecule production on both the transcriptional as well as the protein level, compared to cells that do not have any inhibitor present. And so this tells us that because these pathways are inhibited and it results in a limited pro-inflammatory molecule response, that the PI3 kinase pathway is therefore playing a role in inflammation. The MUC2 knockout cells are slightly different, so on the transcriptional level they also result in a decrease in pro-inflammatory molecule production compared to cells that do not have any inhibitor. However, on a protein level, the cells that have the inhibitor, they demonstrate approximately the same amount of pro-inflammatory molecule production as cells without inhibitor. So this indicates to us that beyond the pathway, there are other mechanisms at play regulating the pro-inflammatory response. So based on the results from these experiments, we can conclude that the PI3 kinase pathway is activated in both wild-type and MUC2 knockout cells, and that the PI3 kinase AKT pathway plays a role in the regulation of pro-inflammatory response in wild-type cells. Moving forward, other pathways such as the IKB NF-κB pathway can be investigated to determine if they are involved in the regulation of a pro-inflammatory response. Thank you for your time.